And so next we have Sarthok Rasique Rahman, who is going to be speaking about the genome-wide DNA methylation patterns in populations from spatial environmental range extremes of the bumblebee, Bombus vosnesenskii.
Thank you so much for the introduction. Hi, everyone. My name is Sarthok, and I'm currently a postdoctoral researcher at Logil Lab and University of Alabama. So today I'm going to talk about recently published work from our lab. So feel free to take pictures and do social media, as we welcome it a lot. So before we go into the details of this exciting study, let's delve into some of the evolutionary and ecological context for the rationale of the research we have conducted. So as we all know that phenotypic traits are determined by a combination of genetic and environmental influences. And if we look a little bit more deeper into these environmental influences, we can actually divide them into two broad categories. One is biotic factors, another is abiotic factors. So these biotic factors include the ecological component like floras and faunas, and also the ecological processes like mimicry and predation that's going on in ecosystem, and also the abiotic factors that includes all the bioclimatic variables such as climate in general, mostly temperature, precipitation, elevation, or the UV exposure, those kind of things. So both of these factors, alongside with the genotype, they actually impact the adaptive landscape and the phenotype of a species. So in this modern age, we are very lucky to have a lot of omics approaches, which we can actually use to examine the impact of these biotic and abiotic factors from the epigenetic level to the coding sequence level to protein and mRNA, and even in the metabolites level. So the goal of our study is to actually use multi-omics approaches to understand the complete picture of this adaptive landscape, which is shaped by multiple biotic and abiotic factors across different levels of biological organizations. So in order to achieve this kind of goal, we always prefer to species widespread species. And why? Because widespread species, they actually live across complex bioclimatic landscapes. And these widespread species, which actually lives across a gradient of altitudes and latitude, they actually face a huge variation in environmental variables within their whole species range. And we can take advantage of this type of natural setup to untangle the evolutionary and the ecological processes that's happening behind the scenes in this complex bioclimatic landscapes. So in this particular study, we actually utilize Sierra Cascades as a natural laboratory for studying the latitudinal and altitudinal effect on species evolution. And in this case, our focal species is Bombus vosnesenskii. So this is a species which is a very widespread species, and it's actually a generalist montane species, which you can see in this map of Western North America. It's living in a huge gradient of elevation, and also the habitat they live, the temperature varies a lot. So they uses a greater habitat diversity at small spatial scales, which actually makes them ideal for the type of study we are actually planning to do. So I'll very quickly go through some of the key works has been done in this species, mostly from actually our lab. So first study I want to mention, which actually looked at the signature of local adaptation in Bombus vosnesenskii. It actually found some very few key candidates, which was actually associated with the temperature or the thermal tolerance of the species. So if you look at the gene list, you can see most of them were actually associated with neuromuscular function, neural development, ion transport, those kind of things, which is very much in line with the other studies found in relation to thermal tolerance. We also looked at the critical thermal limits, and we found in the lab-reared queens that CT mean, or the critical thermal minimum temperature, was varying strongly by the climate of origin, but interestingly, this was not the case for the CT max. Also, we have done RNA-seq experiment studies where we found the most region effect genes driven by the high elevation climates, which is for this study was the Oregon, or actually in the cold treatments. So based on of this knowledge, we actually designed our study, where we actually took a very different approach, where we actually started looking at genomic and epigenetic variability together. And we also wanted to figure out that how this epigenetic variability could be linked with the thermal tolerance of the same species living in two very different climates. So the study design is pretty simple. As you can see, we have two study sites. One was in the Oregon, which we will call high elevation, low temperature site, and the other one was in the California, which is the low elevation, high temperature site. So you can see the number of samples we used for each of the sites for both of the approaches. So we had two different approaches. One was the population genomics type of study to unravel the population structure, and the other one was doing using whole genome bisulfite sequencing to understand the methylation profile, and also look at the general and the differential methylation patterns in relation to thermal tolerance in these two very different bioclimatically variable sites. So first, I would like to go through the population genomics part of this study. So to do that, we have done whole genome sequencing of 18 samples, a very standard population genomics pipeline, which involves sequencing, trimming the data, alignment to the Bombus vosnesenskii genome, variant calling, and using those variants, looking at FST and the nucleotide diversity. So let's go quickly with the key findings from this population genomics part of this study. We found across 18 samples almost more than 1 million SNPs. And the key results I would want to emphasize that we found that Bombus vosnesenskii was genetically very homogeneous. So we found a little bit of population structure, but that was very, very weak. Also, we looked at the nucleotide diversity that was quite similar and highly correlated across the genome between two populations. So we didn't find any clear peak in the Manhattan plots or no clear genetic differentiation, which actually tells us a very important ecological thing, that there is actually a substantial range-wide genetic connectivity between the populations. So the populations are quite admixed. And now I would go to the methylation profiling part. So this is the first of this kind of study which actually look at the ecological epigenetics in the bees. So we first wanted to characterize what's actually the general methylation profile across the whole genome. And then we did the differential methylation part, which looked at the methylation differences between two different thermal populations. So for that, we have done whole genome bisulfite sequencing. That's the pipeline. And then what we had here, that we divided all of the CPG sites into 3 different categories: methylated, sparsely methylated, and unmethylated sites. And we annotated those sites based on their features, like is the site is in the exon or intron or in the genic region, those kind of stuff. And also we have done gene ontology analysis to figure out that what could be the possible functional importance of those sites. For differential methylation, it's pretty much like differential gene expression type of studies, where you actually identify genes. Here in this case, we looked at the differentially methylated sites or the regions at 10% methylation difference. And we also annotated those differentially methylated sites based on the NCBI RefSeq annotation. And then we have done gene ontology analysis. So first, let's go through the general methylation profiling. Interestingly, what we found that general methylation was low across the genome, around 1%. That's actually very consistent with the other arthropod genomes and reported studies, where we know in humans is actually on the other end of the spectrum. You get 70 to 80% methylation in plants, in between 30, 40%, something like that. So that's kind of the general generalization I'm giving you. Also, another interesting thing we found, that transcription start sites of this nearby CPGs were much shorter, the distance, for the methylated sites compared to the old CpGs. And here I have given you a random plot of one of the chromosomes where you can see there's a lot of peaks there. And why they are clustered, we wanted to look a little bit more into it. And what we found when we have annotated them using the annotation for methylated, sparsely methylated and unmethylated sites, it actually tells a very cool story. So here you can see the methylated site, which has more than 50% methylation. They are mostly on the coding regions. And this is interesting because it actually suggests a potential functional role for those sites. And also when we looked at unmethylated CpGs, they are actually mostly on the introns and intergenic regions. So that part, after that part, we wanted to look the difference between the differentially methylated. So looked at the difference of methylation between those thermal populations. So here we found around 2,000 sites in 1,200 plus genes. And very interestingly, most of them were actually hypomethylated in the high elevation samples. And out of those sites, most of them, they were actually also on the exons, as I have shown before. That also suggests their potential functional roles. So when we looked at the gene ontology analysis, we found that the general methylation, the sites with general methylation were mostly related with the regulation of cellular process and cell development, where differentially methylated sites were more associated with alternative mRNA splicing and cellular response to the stress. So this actually tells some possible mechanisms how it actually can be dealing with thermal tolerance in two very different thermally variable populations. So we also looked at the relationship between methylation and population genetic diversity. So we found that the low nucleotide diversity windows had fewer CpG sites in general, and it was actually negatively correlated with nucleotide diversity. In contrast, we didn't see any type of strong relationship for FST and the mean percent methylation difference. So in conclusion, I would like to focus on the key findings that we found the genetically homogeneous Bombus vosnesenskii, no major differences at population level, and methylation results, general methylation results are inconsistent with the other arthropods, but also we found the signatures and potential role of altering gene expression and adaptation to the thermal tolerance via differentially methylated regions. So we are future work or the current work, we are looking at the other species in the same region, Bombus vancouverensis, for the same that we are replicating the same type of study, and we are also collecting RNA-seq and methylome data to look at the links between all of these multiple stages of biological organization. So with that, I would like to finish my talk, and I would like to thank all of our collaborators in this NSF Rules of Life grant. And this paper was just published last month, so please feel free to go and read. There's a lot of details in this study. And also, if you have any questions, I would be happy to answer them right now, or please feel free to shoot an email. Thank you so much for giving me the opportunity to speak in this conference. Thank you.